Hi Tino, I'm Tony from Elena Loisel Lab at the University of Rochester Medical Center. It's a great pleasure to have you today and get an interview and get to know you better.
Nice. Thanks for the invitation. It's great.
So let me introduce you real quick. So you're Tino Stauber, and you are a current master and PhD candidate at the Institute of Orthopedics at the University of Zurich and Department of Healthcare Sciences and Technology at the ETH Zurich. And your undergrad degrees have been Bachelor of Science in Health Sciences and Technology from ETH. And your graduate degree was also a Master of Science in Biomedical Engineering from ETH. And your past employers have been, oh, that's the question. So who have been your mentors in general up until now?
My mentors. So I think I started my science career in the AO Davos together with Professor Claudia Löbel. I think she got her assistant professorship like a few months ago. And I think she was I'm super happy for her. And I think it was important there to, as a first contact with science, to not like frustrate it immediately, and she did a very good job there.
That is great. That's very nice, especially in the beginning.
Yeah. I then continued with Professor Marcy Tsenobi Wong, again cardiology engineering, which was also great. A different set of learning moments, I think.
Oh wow!
Really augmenting, I guess. And yeah, I then moved on to Chess's lab, where I'm still working, which was mostly also like personal development. And I mean, 4 years, 5 years, it's a long time.
Yeah, no kidding. This is nice. So based on all this, experience you had with research and the different labs that you've worked on, what has been your like specific research area and expertise, and especially for tendons, that what you're also working on?
Yeah, I mean, the previous projects were more on cartilage, and tendon was a bit of a shift, but the diseases are not that different, I guess. Same markers or So the start of my PhD was more about the biological response to structural damage in tendon. So quite basic, just like explant models, and see how they react to being overstretched. And that ended in a paper too. And I then realized that maybe for an effective healing study, I need other cells than just those in the tendon core. So I moved the project towards cross-compartmental signaling between cells that are known to be involved in tendon injury or in tendon healing. And this 3D hybrid hydrogel explant model system that I developed during this time has also led to a publication. And yeah, that's pretty much my expertise with flow cytometry. Now I'm doing RNA sequencing a little bit.
Really exciting. I read your paper with the tendon constructs. It was really, really interesting. I really enjoyed that paper. So
thanks.
Great job.
Thanks. I enjoyed writing it as well. So I think
the second version, the next part, that would be nice. So what are you
the next part of the paper, you mean, or yeah?
So I remember you published one in Advanced Materials or something. It was like a really good journal too.
Advanced Healthcare Materials.
Advanced Healthcare, yeah. And then you're doing now with the RNA-seq, it's going to be like the second part or the continuation of the project, something like that.
Yeah, I think that's basically what I'm currently working on, is looking more into the role of IL-6 in these crosstalk mechanisms, in these pathways, which is why I had to do the IL-6, the RNA-seq to like go deeper into the pathways. And this
Very
will be
interesting.
the next one.
Perfect. So, except for, the very interesting research and all the projects that you had, what has been like the biggest challenge that you have had in your research, especially lately, that you're trying to like wrap up and finish with your degree?
There was a lot of, stuff goes wrong.
Oh, I totally feel you.
Sometimes you just have a streak of bad luck, but that happens. But aside from that, I think getting comfortable with like big data was a big step or a big challenge. I think it's just different kind of thinking, and getting comfortable with stuff you cannot really see or touch that was like the big challenge of the last months.
Yeah, especially with that big data, I can totally relate.
Yeah, I you probably know better than me how difficult it is, and but also the nice insights you can gain from it, right?
Yeah, exactly. I think it's kind of a steep learning curve, but after you get that curve, the gain after it's really great. So hang in there. will be very rewarding.
Yeah. That's also what I'm looking forward to in the near future, is like analyzing these data sets and create these nice graphs that I hope will improve my knowledge and the knowledge of the field.
Definitely. Great. So then, after going through all this, research and the challenges and the different people you have to work with, what advice would you now give to investigators who are just starting in the field as like new PIs or new PIs?
I mean, for new PIs, that's gonna be difficult for me to give advice because I think I'm just too young. But for like new PhDs, so what I think I could have done differently is to be more open in the tendon field itself. I think it's a super friendly field, and people are excited to share their protocols and their techniques, and you can save yourself a lot of time and nerves by just asking people for advice.
That's real true. Very important.
Yeah, I don't think we are a super secretive field, and there's a lot of space to grow into. So the danger of getting scooped is like pretty low. I think.
That's true. I totally agree.
Yeah. And yeah, I mean the second thing, I think, as I think we discussed already before, it's normal that stuff doesn't work.
Yep.
Also for an extended period of time, and I think when you start, you just need to prepare yourself mentally that's going to happen, that it's normal, and that you don't need to worry too much about it. It's going to get better over time.
Yeah, totally agree. That's true. Yeah, it's just your fault. Just things sometimes don't work. Most of the times, if you're a grad student, until that one time when it will work and you'll feel like the biggest success ever.
Yeah.
This is great.
That's just something to be ready, and don't think that everybody's the same. The same body.
Exactly.
Yeah, go around it. Yeah.
So besides research, what do you like to do when you're not, in the lab or on your computer analyzing those RNA-seq data? What's your, like your hobbies, and what do you like to do for fun?
I realized going through this process is like I enjoy things that make me not think about lab stuff. This could be like the overarching category of what I consider fun. So mainly sports that put me into this kind of flow state. Basically gymnastics, skiing,
Oh, wow.
free skiing, and climbing, where I can like lose myself in the movement. That's
I think that these are very, very helpful sports to actually force your brain not to think of, like research and stuff, because you have to really focus. And, with the skiing, you don't want to fall down, or with the climbing, you definitely don't want to fall down. So
yeah, exactly.
That's very smart.
you need to focus on something else, so you
Yep.
you can't think about science.
This is
Yeah.
great.
Good breaks, like good reset points.
And what was the last book you read, and whether you would recommend it or not?
I mean, I just started, or I'm in the middle of the Wheel of Time series. I think recently there was also a series coming out about it on Amazon, I think. But yeah, I mean, it's high fantasy, so you kind of need to like that, I think. But if you do, then the first 6 books are incredible. I would recommend it for these reasons. But
perfect. Good to know. Good to know. Perfect. All right, Tino, it was a great pleasure talking with you today, getting to know you better. And I wish you all the best with finishing up, wrapping up, and in your near and later future too. Hopefully I'll see you next time at ORS.
Yeah, thanks for the nice talk and the interview. And
of course.
The same back to you, of course.
Thank you very much.
Nice
yep. Bye-bye.
Bye.